I want you to meet the folks who are attending this meeting today and some who will be presenting. Up in the top left is Mel. She is an Eco-AmeriCorps person who is helping with the monitoring program this year with our stream monitoring that I'll talk to you about. And on the top right is Peter Isles. He'll be talking to you about the cyanobacteria and the algae bloom information. And the left middle is Mark Mitchell. He'll be talking to you about the in-lake monitoring. And I found this really great picture on the internet of four women who are I think Megan might be present tonight. I'm not sure. So we have Megan in the top left, and Megan was an Eco-AmeriCorps member doing what Mel is doing this year, last year, and now she is working at our lab. There's Heather Pembroke with her bright yellow jacket. Heather's going to be fielding questions and helping with the logistics, but she's the section chief of our monitoring program. And there's Kelly Merrill in the black jacket. She'll be talking to you about in-lake monitoring and assessment as well. And then Leslie Matthews, who helps Kelly immensely and does a lot of the background data analysis and any of the work that you might see when you click on to look at the data that comes up from the lake monitoring. And here's me in the bottom left-hand corner, Danielle, kind of your host, at least for the presentation tonight, and then Oliver Pearson, the program manager for the Lakes and Ponds program on the bottom right. And we wanted to ask, because there's so many of you, we love to talk with you and have you introduce yourselves, but because we won't have the ability to do that due to our time constraints, we'd love for you to introduce yourself in the chat. And even before you ask a question too, just so everybody knows who you are, and it helps us to understand where you might be coming from with your question as well. Please, in the chat, if you can, tell us what you love about the lake and what you're concerned about too. And so it may not really mean much to you, but our staff represent different programs and divisions within the Department of Environmental Conservation. So we all work collaboratively, which I think is pretty neat. And they include folks from the Lakes and Ponds program, our monitoring and assessment program, and the watershed plan.
Here you go, baby.
Hey, Heather, do you want to do a mute all?
Done.
All right. So here we are at the beginning of our presentation, the Lake Morey Clean Water Community Presentation. And we will be having 4 different presentations about some of the water quality information that we've been collecting and that we know about Lake Morey. I will be presenting towards the end about stream water quality. Kelly Merrill will be presenting, I guess she'll be the 3rd presenter, speaking about lake water quality and assessment. She's aquatic biologist with the state of Vermont. And Mark Mitchell, who's the Lakes Monitoring and Community Outreach Coordinator, who works for Vermont, the state of Vermont, and also Sea Grant. And then Peter Isles, who's an aquatic biologist with the state, who'll be talking about the cyanobacteria blooms. And the picture to the right is not a picture that was taken in Lake Morey, but it is a picture of my niece, Carmela, and my sister. And we are out on one of our local lakes where I grew up in near Charlemont, Massachusetts, at Savoy State Park. And so water is really close to our hearts, and all of us, this is what we want. We want clean water so we can have it for, future generations and on and on. And I also want to give a big special thanks to the Lake Morey Commission and the town of Fairleigh for the in-lake and stream sampling efforts. You can see to the right, Greg Allen doing his back-breaking work, collecting water quality samples at all the tributaries around the lake. He's been doing that for 3 years. The support from our Eco-AmeriCorps members. And then you might recognize Don Weaver to the left, who has been active in all things water quality with Lake Morey and the state for probably 20 plus years. So I thought it might be helpful to start with community actions for a healthy lake. And one thing that I think is really important to know about Lake Morey is that they have been very active and doing things to help to contribute to the health of the lake. And that's what we've seen as we've collaborated with folks through our state programs. So the first two steps you see here, collaborate with state scientists and establish a lake monitor. You can see there's check marks above those that are filled in with black. And those check marks filled in means there's a really strong collaboration, a strong effort. And you'll see some ones that are not filled in, but those mean we've sort of been beginning this. We're in the process, but we're going to continue to do that work. So the first two steps are, it really is all about the volunteers stepping up for the long-term water quality sampling through the lake monitoring program. So this has been active at Lake Morey for over 25 years. And this long-term monitoring that happens every year in the summer can identify if pollutants are going up or down, if they're staying stable. And then these trends are displayed on what we call the Vermont Lake Scorecard that Mark will talk about a little bit later in the presentation. In the case of Morey, it's the summer volunteer data that caught the eye of the Lakes and Ponds program. So to determine if there were any obvious sources of pollution from the streams going into Lake Morey, volunteers began tributary sampling through the La Rosa program. So that's the work that Greg's been doing. And Heather and Mel and Megan manage that group. And then cyanobacteria monitoring was also employed. So anyone who is interested can be a cyanobacteria monitor. The word may sound kind of freaky to some people, but Peter will talk to you more about that and what that means. If you can see pretty well, it's something that you can do. And it's good to have multiple monitors at the lake. It's really simple. It doesn't take much time. It's definitely a way that you can be active in caring and reporting for health on the lake. So our staff also prioritizes Morey for annual spring sampling and for 2-day, what we call the next generation lake assessment that Kelly will talk to you about. So it's a larger way of looking at the watershed and the lake. And we've seen evidence that there was a treatment on the lake to help these cyanobacteria blooms that were happening in the past. And we're seeing that maybe this treatment's reaching its maturity possibly. We're not sure. We're thinking maybe there's sediment, at the bottom of the lake that's releasing some of this extra food for these bacteria, and that even the tributaries, the streams that flow in, could be contributing pollution as well. And that lakeshore development could also be providing some of this pollutant. But overall, good sign is that the water quality still ranks good. And Kelly will talk a little bit about that. And then additionally, the state developed this land cover. So it's basically like looking down at the lake and saying, what is around the lake? Is it houses? Is it roads? Is it forests? And based on what it is, that affects like what happens with the water when the water flows through those areas. And do they take water from those areas and contribute pollution, or do they take water and filter it? And so what we've seen with this mapping is that the most concentrated area of development where you can have runoff that captures pollutants is within 100 feet of the lakeshore. So it's this area around the lakeshore that Kelly will talk about. And this equals about 9 acres of this type of surface that can contribute pollutants around the lakeshore. And to give some context to it, if this was one landowner and they had 9 acres of this land, which could be a road, a house, anything that doesn't infiltrate water, suck in water when it rains, they'd be under a stormwater permit. So they'd have to have a permit and they'd have to do all these things to make sure that the water that entered the lake was clean. But these same permits don't apply to land under an acre. So in that case, we really need to rely on a program like Lakewise to help landowners at their small, postage stamps around the lake to reduce that runoff and improve aquatic habitat. So that area that's really close to the lake shore that provides shade and food for all the things that live in the lake. So communities also initiate resource assessments, and these are like really basic things. So it's like looking at your roads and saying, are they in good shape? Are we noticing that when it rains, a lot of the dirt from the road ends up in the lake? Looking at woods roads up in the forest and saying, when it rains, are we seeing a lot of erosion to those roads? You know, are they starting to fall apart and we're losing the surface area? And so that's another thing that we can do, looking at what's going on with water and clean water in the lake. And as part of these lake-wise assessments that I talked about, communities can hold these things called septic socials, which sound like really exciting, right? But Lake Morey has hosted this septic social on site at a new system that was installed a few years ago. And this was before COVID, so everybody was able to get out at the site. And it basically just is a way for people to look and see how septic systems work. So how they collect our waste, you know, and from our houses, from our tubs, our toilets, and how they clean them out so that that dirty water doesn't end up in the lake. So when the lake needs a holistic approach for the entire watershed, so that's the entire area that drains all to the lake, you can develop a lake watershed action plan, and I'll talk a little bit more about that. But they look at all the information around the lake. They prioritize and create projects, you know, like where to fix a road, where to install like a way to capture rainwater that comes from a roof instead of sending into the lake. And they also help us to identify funding sources to support that work. And most important,
sorry to interrupt.
Yeah.
We're at 4:15, so if you could wrap it up so that Mark could start, that would be great.
Yeah,
Okay.
It's common. So the community then implements those practices, and then they continue to track and monitor that response. So all this work ties into the 3 C's, and that's community, which is strong involvement and strong vision for the future of the lake. Commitment, so that's implementing those practices that improve water quality and protect habitat for the future of the lake. And then continuity, which is building lake awareness through education and stewardship. So I'd now like to introduce Mark Mitchell, the Lake's Monitoring and Community Outreach Coordinator, to talk with you about the long-term in-lake monitoring and lay monitoring program.
Great, thanks, Danielle. Hi, everybody. I think quite a few of you are probably familiar with me. I coordinate the Vermont Lakes Lay Monitoring Program, and that is with Greg Allen and Don Weaver. So that's collecting lake water quality samples throughout the summer on about a weekly basis, aiming for at least 8 samples per summer. So I'm going to start with the lake scorecard here that Danielle mentioned before, which is kind of our way to put together our monitoring data and look at trends and status and do some assessments from that data. So the summer volunteer monitoring data are the graphs starting on the upper right here, which is the summer TP, which is total phosphorus. That's what we collect water samples for total phosphorus, and chlorophyll A, which is the graph below, which is a proxy for the concentration of algae or cyanobacteria in the water. And then to the left of that is the summer Secchi annual mean. So that's Secchi depth, which is a measure of water clarity. So if we start at the top right, that's the summer total phosphorus. You can see back in the 80s, you know, it was quite high readings, and that's when we were having algae and cyanobacteria blooms. And that led to the alum treatment in 1986 that I mentioned here at the top. And basically right after that, the phosphorus levels came down as the alum was able to bind the phosphorus into the sediment where it was being released during the summer under anoxic conditions. So that's when the lake thermally stratifies. You have a warmer layer on top, colder layer on the bottom, and they kind of get cut off from each other. So you lose oxygen over the summer, and then that leads to phosphorus being released from the sediment. So we've seen, you know, the phosphorus level has been reduced for years, you know, decades now, some really good monitoring data, with some ups and downs over the years. And some of that could have been from weather, you know, variation, increased precipitation, for instance. Of course, we do have climate change and global warming happening as well. And in recent years, there's kind of been a little bit of an uptick as well. And so that's kind of leading to an increasing trend overall, which is what this graph is showing. with the red, and we have a statistically increasing trend. So it's not a huge magnitude, but we are seeing that. So yeah, that led to some of the concerns to do some additional monitoring in the tributaries, as well as kind of look into the internal loading aspect where the phosphorus is, released from the sediment and how that's holding up after the alum treatment, which has now been 30 plus years, which is quite a long time, but still seemingly pretty successful when you compare the data to the 80s. Chlorophyll, we kind of seen the same pattern with some more recent upticks, increasing, but still relatively low. The colors in the background just relate to trophic state, or basically how productive a lake is, how old the lake is. So we're kind of in the mesotrophic range, which is sort of the intermediate range, which kind of makes sense for the lake depth, about 13 meters. So you have some of these physical characteristics on the left here. And then the Secchi depth is, something to focus on as well, because since the treatment, it really increased and you're getting almost, towards the bottom of the lake. So these are really high clarity values, between 8 and, almost 10 meters. And that's changed over the years a little bit too, but we're still getting really good clarity, up until this year, including, and I should mention this lake scorecard is only through 2020. It will be getting updated shortly as we have all the monitoring data in from last year. And so, we do consider the lake stressed for phosphorus, as the levels are a bit elevated compared to a lot of our other lakes. But, we don't consider it impaired because certainly has improved since the 80s. And, we're getting really good clarity readings as well. So I'll move on to the next slide. Danielle, are you controlling the slides?
I
Sure
can if you want, Mark. I think sometimes when you have the, whatever it is, the laser thing, it makes it hard to move them forward, but I can do that if you want me to.
Okay. Yeah, that'd be great, because I can't see there it is. All right.
Yeah.
Cool. So this is the 2021 monitoring data from the lay monitoring program, as well as the spring phosphorus monitoring that Kelly and Leslie do. I'll let them talk more about that, but that's a sample right after ice out. So if you look at the table here, for the means, again, the summer total phosphorus is a little bit elevated this year, kind of following the same pattern. It's not super high. There's some variation. So the graph on the bottom shows the individual sampling events. Pretty steady. So the phosphorus is in the green triangles here. Chlorophyll is in the circles. And those are pretty low readings, which is good to see this year. And the Secchi disk on the bottom here is sort of how deep that clarity is going. So it kind of increased throughout the summer. There's one blip here in the phosphorus, which could just be from the lower lake levels and the sampling actually reaching further down towards the sediment. And so the way the samples are collected are, we take twice the Secchi depth normally, but in this case, that would always hit the lake bottom. So we're basically doing a whole water column using a hose. So collecting some phosphorus that might be coming from the sediment as well. So yeah, numbers are kind of where they have been the last few years, except, the Secchi is looking really good and the chlorophyll was quite low this year. So that's positive. And then the next slide, let's see. There we go. So I skipped one. And just to give you some idea, comparing to other lakes in the volunteer monitoring program, which we have close to 100 lakes, including some Lake Champlain stations. So for the Secchi, as we kind of go to the right here, which is higher clarity, Morey is actually right up there with the top lakes. Some of our really deep oligotrophic lakes, like Lake Willoughby, for instance, Maidstone. So that's really good to see, which is kind of why we don't consider the lake impaired for aesthetics or recreational value. And just on the bottom here is Don Weaver. And here's a picture of a Secchi disc. So when I was talking about, we lower this into the water until we can't see it anymore, and that's the clarity. Excuse me. And yeah, so moving on to, oops, wrong way. Chlorophyll also this year was quite low on the lower end of the spectrum. Actually, kind of what we would consider oligotrophic, or a very clear unproductive lake as far as plants and algae. So that was a good result as well. But when we move to phosphorus, it is on the higher end of the spectrum, this year especially. So that does give us a little concern, but part of that is the sampling methodology, which I'll get into in a second. And we see fairly, just as a comparison, a little bit, I think around the same. It's actually a little bit lower, kind of in the middle here, but Morey does have higher clarity readings that we've been getting than fairly, and a little bit lower chlorophyll. So this is some of the additional sampling I've been doing when I've come out to do lake visits with Don and Greg. And this is to kind of get some information about internal loading and what the status is with that. So I bring additional equipment. I take lake profiles, so going down vertically through the water column using sensors, so it's a probe, as well as taking surface and near bottom water samples for comparison. So what we're looking at here on the left-hand side is the lake depth going down to about 12 meters. So I sample down to about 1 meter off the bottom. The black circle here is the Secchi depth. So that day was quite high, just over 8, and this is July 6th of this past year. And then the red line is temperature. So that's showing the stratification in the summer. So warm water on top. So you have increasing kind of numbers or concentrations here. And then there's a mixing layer in the middle, and then it goes down to a bottom colder layer known as a hypolimnion. And you see this pattern with dissolved oxygen being fairly high at the top. There's a bit of a spike here in the middle, and that's likely where cyanobacteria and algae are kind of hanging out. So they're producing some oxygen, which is quite common to see in lakes in the summer, kind of hanging out in that middle layer. And that's where the Secchi depth is basically going down to. And then it drops drastically down to near the sediment. So you're still having some anoxia or low to no oxygen. And that can lead to that internal loading of phosphorus that I mentioned. So what I did was take a near surface total phosphorus reading, and this is quite low, below 10 micrograms per liter, as opposed to, the 26 that we were getting for an average. And that's why, you see this clear water at the top, especially when you're swimming or boating. But then towards the bottom, it increases, it doubles, basically. And I'm about 2 meters off the bottom here. Compared to the hose sample that day taken by the volunteers, was actually a little bit higher. So it might have went even further than I actually took my sample, because the hose is about 10 meters plus or minus, depending on the lake water level here. So, we're seeing the bottom sample affecting the hose sample, but is that really indicating how, what the aesthetic value of the lake is? Probably not, because we're really just looking at these upper layers, the upper layer. And dissolved phosphorus, kind of the same sort of patterns. It's very low, but it does increase towards the bottom, which you would see with internal loading. And I just have a couple more graphs here showing the exact same thing from 2018. The same kind of pattern. Here's the hose sample. It's being influenced by the bottom phosphorus. Near the top, it's very low. Good clarity, again, over 6 meters, and low oxygen at the very bottom. This is a chlorophyll sensor I had working. It kind of shows an increase in cyanobacteria and algae in that kind of middle layer. And cyanobacteria can actually move up and down the water column. They can pull phosphorus from the bottom and then kind of come up and bloom, typically during the fall turnover, but it can happen during the summer as well. Oops. And I'll just show one more graph. This is late summer, so it's actually after the normal volunteer season. 2018 I came, and you can see a huge increase in the phosphorus here, and that's as it builds up over the summer. Especially with some global warming, we might have longer periods of stratification of the lakes, and then more depleted oxygen levels and increased phosphorus from the sediment. So we're definitely getting some internal loading. I still don't think it was nearly as bad as it used to be, just because of the numbers being quite a bit lower. And one thing I should mention too, so iron and manganese I also collect, and that's an indication of internal loading, as that binds to the phosphorus in the sediment, and then is released when it's anoxic under those conditions. So we look for increases in iron or manganese towards the bottom, and in this case, it's really the manganese that's the increasing factor here that's bonded to the phosphorus. And this was under the ice. I did once as well, and I took a few more samples in the water column. So there's some variation in the water column, but still quite low until you got down to about 1 meter off the bottom. So there's definitely some, internal loading, I would say, happening in the winter as well, when there's low oxygen levels. So it's something to keep an eye on. And with that, let's just talk about what we're looking to do this upcoming season as far as additions to the lake monitoring program. We'll be trying to take lake depth. I think the Association of the Volunteers have purchased or are going to purchase a handheld sonar, which I have one now as well. That'll help know, how deep your samples are actually going, so we're not hitting the sediment, and if the lake water level is actually changing. And then a second YouTube, I've gotten all the volunteers. I should be able to equip all of them this summer with these. This is what Greg's using right here, which really helps with a more consistent Secchi depth reading. And then we'll do surface bottle grabs here because there's some indications of internal loading. So we'll do that for total phosphorus, chlorophyll A, and I'd like to add caffeine as a new parameter that the lab is testing for, which can indicate wastewater, especially septics around the lake. So that would be some good baseline information to gather. And then, cyanobacteria monitoring, while they're doing the sampling would be very helpful. And with that, I think my time's just about up. And I guess we're accepting questions now if we have time.
Mark, there was just one question about when the septic social was from Jim Chambers.
Septic social, I'm not exactly sure when that would have been. I'm not sure if Oliver or somebody else would know, because that was probably with Amy Peacock.
Yeah, I think Mark and his partner are on the line. Maybe they can type in the chat or say out loud when they did that.
It seems like this is Mark. It's been a couple years anyway, but we're trying to remember when exactly it was, but I can't tell you for sure.
Thanks.
But I guess Kelly is next.
So I've gone ahead and shared my screen. Can folks see that?
Yep.
All right. So I'm Kelly Merrill. I'm currently a select board member in Danville, and before that I served on the Planning Commission. So thank you to those volunteering here tonight on behalf of the town and Fairleigh and Lake Morey. I'll try to make this presentation a little more engaging than some of the ones I know you get to sit through. I'm an aquatic ecologist with the Vermont DEC's Lakes and Ponds program, and I have had the privilege of getting to monitor the status and trends of Vermont's inland lakes since 2001. My colleague, Dr. Leslie Matthews, and I sample roughly 70 lakes each spring, attempting to hit them at spring turnover when the lakes are fully mixed, right after the lakes ice out. And that spring sampling program is going since 1977. And while we take a variety of water quality measurements during the spring, the Vermont Lake scorecard shows the graph for only one of those parameters, total phosphorus. And as Mark had shown using the Vermont Lake scorecard for Lake Morey, we're not yet seeing that increase in total phosphorus in the spring, but we are seeing that significant increase in the summer. As measured by the volunteer lake monitor, and it's statistically significant. And when we see a lake with an increasing total phosphorus trends, that puts it on the priority list for my colleague, Dr. Matthews, and I to visit it in the summer for a comprehensive 2-day checkup. So this is only a one-time visit, but it's a visit where we look for possible sources of phosphorus pollution at the deep hole, what we call the index site, at 10 random sites around the lake using the U.S. Environmental Protection Agency's protocols for assessing physical habitat health, and by sampling all the tributaries that we can find and looking for signs that there may be sources of phosphorus upstream. So last summer on August 24th, we sampled the deepest point in the lake that we termed the index site, which you can see in the location of this map. Claire Potter reported for the Valley News, and photographer James Peterson came out with us while we were sampling that day, and you may have read their piece in the Valley News. Now, unlike spring P, and in the spring and in the fall, when the lake is fully mixed, in the summer, the lake stratifies. And it's warmer at the surface than it is at the bottom. Anyone who's dove into a lake in the summertime and gotten down to that chilly water down below has experienced getting into what limnologists call the hypolimnion. So this is a graph from our visit. And what is shown here is the depth. This is the surface of the lake up here. This is the bottom of the lake down here. This blue line is the temperature profile. You can see in the surface waters, they're much more warmer than in the bottom waters. And actually, where there's the biggest change in temperature is called the thermocline, and that's noted by this dotted line here. And so what happens is once in the summertime, this stratification sets up, this bottom cold water doesn't mix with this warm water up here. And so basically that sets up a scenario that makes this water down here different from this water. And so if we look at this green line here, which I'll move to the next one. If we look at this green line here, this is a measure of algae productivity. And what we see in the surface waters is there's not that much, even though we were there during what we think was a Gliotrichia bloom, which is a form of cyanobacteria. There really wasn't very high concentrations in the upper water column. But once you got down below the thermocline, we had some very high levels of this Gliotrichia is what we think we were measuring. And what happens then is that so anyway, I'm guessing that Lake Morey has a lot of snowbirds, folks who live on the lake in the summer but go somewhere warm and sunny in the winter. Now, the interesting thing about Gliotrichia is that it's a bit of a snowbird as well, except instead of driving back and forth to Florida, it goes up and down in the water column. And going down to the cold, darker water, it fills up on hamburgers to fatten up in the cold, and then floats back up to the surface to relax in the sun and photosynthesize like your houseplants. Except algae isn't eating hamburgers, it's taking up phosphorus, which, as Mark said, is in higher concentrations in these bottom waters because as these algae die, they settle into the bottom and they start to decay, and the decay uses up oxygen, which we see in this black line here, that there's no oxygen at the bottom of the lake anymore because the bacteria that are eating the dying algae are using up all that oxygen. That causes the chemical reaction, which releases the phosphorus from the sediments. And then the Gliotrichia go back after they've been having their hamburgers up into the surface waters where it's nice and light, and they bring some of that phosphorus with them, which is why we see this higher level here at about 4 meters, where there's more light and warmer temperatures. So the sediments would otherwise be, the phosphorus that's in the sediments came from the drainage area of the lake at some point in the past. And so, as Mark mentioned, we found further evidence that the sediments are releasing phosphorus by the measure of total manganese. So when we were there, it was total manganese in the surface water was very, very low, it was 8 micrograms per liter, but it was super big in the bottom waters. All that you need to know about this is that this indicates that the sediments are probably releasing phosphorus, feeding some of these algae blooms. But before we move on to the tributary and habitat data sets, I just want to put Lake Morey's water quality in context for you. Across the board, no matter what water quality parameter is used, Morey scores good for water quality. Other lakes across the Northeast and the nation do not score as good across all these parameters. So while we are seeing trends that are of concern, the current water quality of Lake Morey is still excellent when compared to lakes across the region and nation especially. Morey's surface algae concentrations, even though we were there during an algae bloom, are in what was considered mesotrophic. And that's denoted by this light blue color. And you can see, like in other parts of the country, there's not many lakes that are in as good water quality as Lake Morey is. Now, I'm going to point out a couple of things worth sharing about the observations and measurements we made at the inlets during our visit last summer. We sampled the 9 tributaries shown here with the blue lines. The red dots show the amount of total phosphorus measured. The bigger the dot, the more phosphorus that was measured. Tributaries 8, Bonnie Oak Brooks, and tributary 9, Pine Brook, had the highest concentrations of total phosphorus. In this view, we see levels of conductivity shown in these big white circles. Now, conductivity can be a proxy for pollutant runoff. You can see that inlets 1, the Aloha, and 8, Bonnie Oaks Brook, have the highest conductivities, as well as the highest total chloride readings, suggesting that road salt is running off into these inlets and then into the lake. 2 milligrams per liter is the detection limit and represents what we would expect in total chloride concentrations if there was no road salt influence. We see that for inlets 2 through 7, we are getting such low concentrations of chloride, suggesting road salt runoff is not a problem in those tributaries. Now, the pink dots, which you don't see very many of, just one here, denotes the size of any deltas we observed. Now, some lakes have really big deltas on it, like Lake Carmi, Lake Willoughby, but we just observed one at this one inlet, the inlet number 9, or Pine Brook. It wasn't very big, but you can sort of see the sediment there, suggesting that upstream there's some sort of runoff happening. Now, here are the turquoise dots represent percent cover of algae that was observed at the inlet mouth. So this can indicate that nutrients may be entering at an inlet with a lot of algae growth. At none of the inlets did we see 100% cover of algae, which we've seen in Shadow Lake, in Glover. But at inlets 5 and 6, we saw between 40 and 60% cover respectively, suggesting maybe worth looking upstream of those 2 tributaries as well. So all of this tells us that there are a couple of streams worth looking upstream of. Inlet 8, Bonnie Oak Brook, and had a high enough total phosphorus and had evidence of road salt runoff. Inlet 9, Pine Brook, also had high total phosphorus readings and the largest sediment delta, which wasn't really that large. And then Inlet 1, Aloha, had evidence of road salt runoff. And then inlets 5 and 6 had the highest percent of cover of algae, but still wasn't very big. So I've shared with you the highlights from the Open Water Index site and the tributaries. Now I'm going to share with you an assessment of the condition of the lakeshore, which can also be sources of runoff and pollution to a lake. This approach uses the same approach used by the US EPA National Lake Assessment, and we visit 10 randomly selected sites shown here by the letters A through J. And at each site, we make observations that get plugged into 4 of EPA's indices shown on the right. The orange-brown circles show how embedded the sediments are off of the sites. Embedded sediments indicate that there is runoff of sediment happening that is resulting in the burial of rocks. So as you can see, we had some measure of embeddedness off these sites. Now recall how well Lake Morey scored for its water quality parameters. Here we see Lake Morey is not scoring as well for the condition of its lakeshore. It scores fair for lakeshore disturbance, which is a measure of the intensity of development on the lakeshore. And it scores fair for lakeshore habitat, which is a measure of the tree canopy, understory, and the ground cover, and then the spongy duff, which absorbs rain and slows rain down. And then it scores poorly for shallow water habitat and poorly for its physical habitat complexity, which is a combination of lakeshore and shallow water habitat. You'll recall that when we sampled the tributaries, we noted if there was any algae present, which can indicate phosphorus runoff upstream. Interestingly, we didn't really see evidence of that at most of the tributaries. However, we did see evidence of a lot of algae growing close to shore off of this developed site. which has a slope with no tree canopy, no understory to slow the rain, and no spongy duff layer found in woodlands that absorb the rain. So rain just washes down this slope, bringing sediment and nutrients with it, and potentially fueling the nearshore algae growth we see shown in the picture on the right, which is right off of that. So now I want to share with you a new tool we have to use to help home in on the possible sources of phosphorus as well. Vermont is fortunate to be the first state in the nation to have very high resolution land cover down to 1/2 meter. Most other states have it only to 30-meter resolution. So as a result, the Lakes program, with help from the IT branch of the state government, was able to develop land cover summaries of the watershed, the air drainage for the whole area that drains to the lake, the tributary buffers, and lakeshore buffers. And if we look at the summary for the watershed, that area that drains the water to the lake, we see that only 1.7% is impervious surface of roads, roofs, decks, driveways, and only 0.6% is agriculture. But if we look at the land cover for the 100 feet on either side of the tributaries draining into the lake, we see that it's a little bit more. There's 2.6% is impervious and 0.4% that's less agriculture. And you'll note that the National Hydrography dataset does not show tributaries Bonnie Oaks or Pine Brook, which we measured the highest phosphorus readings at.
Hey, Kelly, sorry to interrupt. You just have a couple minutes.
Okay. If we look at the land cover within 100 feet of the lakeshore, we see that 15% is impervious and 5.5% is mapped as agriculture. This shows that impervious surface is in the watershed is concentrated within 100 feet of the lakeshore around Lake Morey. There's roughly 9 acres of impervious surface located within 100 feet of the lakeshore. So if this was Lowe's box store, they would have to come under stormwater regulations that require the reduction in phosphorus runoff to the lake. But it's not one landowner, it's many landowners. So stormwater regs don't apply. And we must rely on programs like the LakeWise program to help landowners collectively do the right thing. Ultimately, our visit last summer doesn't really tell you much you didn't already know. Phosphorus seems to be coming from 3 different sources: the sediments in the open water, the dissolved oxygen in polluted areas, the tributaries, and from the lakeshore development runoff. Moving forward, and work in any of these areas will help reduce the phosphorus load to the lake and help turn the increasing phosphorus trends around before it's too late. And finally, I just want to make you aware of another statistically increasing trend we are seeing on Lake Morey. Total chloride, which is a measure of road salt pollution, is significantly increasing in the lake. If there was no road salt, we would expect a concentration of 2 milligrams per liter. But as you can see, when we first began measuring total chloride in the spring turnover of 2007, it was already at 10 milligrams per liter. And as of 2021, it was up to 17 milligrams per liter. There are only a handful of lakes in the state with a statistically increasing trend for total chloride, and Lake Morey is one of them. A new study out of Dartmouth and with many other collaborators has found that when concentrations get to around 50 milligrams per liter, zooplankton, the microscopic organisms that eat algae, they die off. So more road salt, less zooplankton to eat algae. So presumably we'd see more algae in the future if we don't work to reduce the road salt runoff to the lake as well. So if you'd like to explore the interactive map report that I just presented here, you can email me for that. We hope to post that to the website sometimes this summer. And if you would like to explore the land cover more closely, you can find that now on the web. Thank you.
There was a couple of questions that came up in the chat. Ted Fairley, town administrator, asked what was the perimeter line around the lake?
The yellow line. I think it was a slide before it. I just was curious. Is that the watershed? If you go back a couple of slides, I think. Yeah.
Yes. Yes. Sorry, I didn't. That is the watershed, and it's here. That's on the scorecard. And that shows you the area that that drains to the lake.
Another question from Arlene. I'm wondering if the sediment loading varies in different areas of the lake. Like, is it high in the north end, or are you only sampling in the deepest part of the lake?
That is a good question. You know, we didn't really see, like I mentioned, at the tributary mouths, often you'll see these deltas where there's sedimentation happening. We didn't really see that as much as we see it on some other lakes that we've sampled around the state. This is a measure of that embeddedness, so presumably it's a measure of how much runoff. I think this is maybe a steeper side of the lake too, so it would make sense if there's any clearing on a steeper slope, you would have more probably runoff, which is burying the sediment. This is a measure of how buried sort of cobble that we find is, and how much that interstitial space where macroinvertebrates like to live gets buried by runoff. So not sure if that answers the question.
Yeah, Kelly, I was thinking she might be asking about the internal loading, and Peter had a response to that he'll get into that with his presentation as well. But yeah, it could vary in different parts of the lake. And we just sampled the deepest part as kind of the most integrated area of the lake, but it's going to be related to the low or no oxygen levels at depth. So it just kind of depends on how deep and where that oxygen depletion is happening.
We have a couple more questions, but I'll keep track of those and we can ask them at the end. I think Peter Isles is up next.
All right, thanks, Heather. Let me try this. Sorry, waiting. Here we go. All right, are you guys seeing this? Are you guys seeing the
Yes,
full
we're
screen
seeing.
presentation? Or are you seeing my notes?
Seeing your the slideshow.
Okay. Sorry, I'm a newbie at the SharePoint PowerPoint presentation.
And I can always share the screen for you, Peter, and give you control too, if that's easier.
I think it's okay. It's just taking a little while to load all the slides and find my place.
Okay.
Here we go. All right. Sorry about the delay. So my name is Peter Isles, and I am the project leader for the cyanobacteria monitoring program in Lake Champlain and throughout the state, as well as Lake Champlain long-term monitoring program. But today I'm going to talk a bit about cyanobacteria in Lake Morey. Just so you don't get your hopes up too much, we don't have much data on cyanobacteria on Lake Morey, so I'll kind of present a lot of information about our monitoring program and how we do it, and then some features of Lake Morey that might have a bearing on what cyanobacteria in the lake are likely to do, and then just make some educated guesses in terms of what might be going on in terms of cyanobacteria ecology in the lake. So what are cyanobacteria? Let me see. Cyanobacteria, you might know as blue-green algae. So they are unicellular, often colonial bacteria capable of photosynthesis. So they're a form of what we call phytoplankton, which are basically functionally microscopic plants living in the lake. They've been around for a long time, and they live in almost every environment on Earth, and they're a natural and important part of most ecosystems. So on their own, cyanobacteria don't necessarily indicate that the system is in trouble. But of course, when you get too much nutrients in a lake, then runaway growth can lead to blooms, which can be toxic and can have other ecosystem consequences, in which case that can be problematic. In terms of what kind of cyanobacteria are in Vermont, these are some of the main problem species. So we have Microcystis in many lakes in the area. This is one of the most common problems, cyanobacteria, around the world, and it can form dense blooms like you might have seen on Lake Erie in the news. In a lot of deep lakes, where you have these deep chlorophyll maximum, like Mark and Kelly were showing earlier, you have blooms of Planktothrix. Not so much in Vermont, but it's something that could potentially like that type of habitat. So these tend to live in a deeper layer of the lake, but they can cause problems when they mix up to the surface waters. So far, we haven't seen that much around here. We have Dolichospermum and Aphantazomenon and Gloriotrichia. These are all nitrogen-fixing species. And of these, Kelly mentioned that Gloriotrichia has been observed in Lake Morey. And as she was saying, this is a somewhat unique species in that it often appears in lakes with relatively low nutrient concentrations in the surface waters because it's really good at storing up nutrients from the deep waters and then floating up to the surface and living off those stored nutrients for a big part of the summer. So how do we recognize cyanobacteria blooms? I mean, this is an important thing for everybody around the lake to know. And basically, they can look like a number of different things. They can be relatively sparse, like you see on the left here, just a few dots and you're kind of floating on the surface, but not forming, a continuous layer. I see a question from Bob, which I'll try to touch on in a moment after I go through these slides, about aerosolization of the toxin. You can also see these blooms concentrated in a specific area and getting much more dense under the right conditions. So what tends to happen is that these cells float. They have little gas vesicles, basically gas bubbles that allow them to come up to the surface, and then the wind can concentrate them in a certain area of the lake. And that's when you get the highest concentrations of cyanobacteria and the highest chance of exposure to toxins. There was a question in the chat about aerosolization of toxins. Is everybody else seeing the slide? Sorry, I see somebody does not.
We
Yeah.
can see.
Okay. Basically, there's not enough data about aerosolization of toxins to say much. They have been measured in aerosols, but there's no frame of reference for those concentrations and whether they would be anything dangerous. There's also discussion of something called BMAA, which is a type of amino acid that's been implicated in neurodegenerative brain diseases like ALS. Again, I think the jury's still out on the science, and I think the position of the research community is that it's too early to say that there is a link there. So let me continue here. So how we monitor cyanobacteria blooms in Vermont is mostly through visual assessments, like you're seeing in these pictures. So what we count on is volunteers going out to the lakes, preferably at the same time every week, and making an observation. Do they see a bloom? And is it a big bloom, a small bloom, or is it clear water? So it's really important that we have people who go out in conditions when there's no blooms and make reports. It's also important that, of course, we report as many blooms as possible. As of now, we don't have a lot of those reports from Lake Morey, and it would be great to get more information about that, particularly as there have been some blooms observed. It's also important that you guys know what is not a bloom in Vermont. So we get a lot of calls about things that people think might be blooms, and not everything is. So something that people often ask in the spring is concerns pollen. So you see these thick accumulations of yellow pollen on the water, and this can look like blooms to some people. But generally speaking, if you see this in the spring, around the time that the trees are releasing their pollen, there's probably pollen at that time of year. Generally speaking, if you have filamentous algae growing on the bottom of lakes, these tend not to be cyanobacteria. They're usually other forms of algae, and these can also become detached and form clumps that float in the water. And again, those are not necessarily cyanobacteria blooms. And finally, duckweed is a small flowering plant that can look like cyanobacteria from a distance. But if you get up close to duckweed, you can see these 4 little leaves on it, and those are structures you would not see in cyanobacteria because the cells are much too small to see with the naked eye. To be even more sure whether it's cyanobacteria, the best thing to do is to take a jar of water from the lake, you can put it in front of a white background and then you can start to see the particles inside. And if you're going to report it to us on the website, it's very useful to see those photographs. And if you leave that jar sitting for 20 minutes or so, you start to see those cells in the jar floating up towards the surface. This is a good indication that it's probably cyanobacteria, because as I mentioned earlier, those cyanobacteria have these little gas vesicles inside, which makes them float under calm conditions. Generally speaking, if it's algae that you can pick up with a stick, it's probably not cyanobacteria. This is not universally true, but for the most part, it's a pretty safe rule of thumb. So if you can pick it up with a stick, it's probably not cyanobacteria. And finally, when you make those observations, we encourage you to report those to the cyanobacteria tracker, and this is maintained by the health department. So you can do this as one of our trained volunteer monitors, or you can do this as just a citizen, and we have different web forms to do that, which you can find if you go to this tracker website, or you can reach out to me at any point. So when you enter information about the tracker, then some of us in DEC, in the health department, or at our partner Lake Champlain Committee will take a look at the photos that are submitted along with each report and just vet that they really are cyanobacteria blooms or likely to be cyanobacteria blooms. And then those will show up on this map in real time with maybe up to a day or so delay. So you can see where blooms are happening around the state during the summer season. All right, so that's just a bit of background about blooms in general in Vermont. In terms of specific questions to Lake Morey, I think it's useful to be aware that cyanobacteria blooms used to be a pretty common occurrence. I don't know of any data on that, but they're reported, at least in this paper by Eric Smeltzer, which was kind of led up to the alum treatment that occurred in the 80s. So a study at that time determined that internal loading was the major source of phosphorus to the lake. And it was assumed that a lot of this phosphorus had been delivered decades earlier as a result of poor wastewater management practices. So because it was determined at that time that it was probably historical inputs of phosphorus and not ongoing inputs of phosphorus responsible for the problem, that was why they decided that alum was likely to be effective in this lake. And it did seem to be, as you could see from the figures that Mark showed earlier. However, it does seem that it may be increasing again. And I think this might be partly because Morey just naturally is susceptible to internal loading. And I should say again, what I mean by internal loading is the release of phosphorus from lake sediments. So normally speaking, phosphorus binds really tightly to clay particles and to iron and manganese compounds when in the presence of oxygen. But when there is no oxygen, then iron and manganese dissolve through a process called reduction. And so any phosphorus that was sticking to the surface of those particles can pop off and float up into the water column. So this is why those manganese readings that Mark and Kelly were showing earlier are indicative of internal loading, because it means that oxygen is low enough to dissolve those iron and manganese compounds and release phosphorus. One reason why I think there tends to be some internal loading issues in Lake Morey is because the bathymetry of the lake is somewhat unusual in that you have these quite steep sides to the lake and then a relatively flat bottom area. This means that almost all of the lake sediment surface area is exposed to anoxic conditions under some summer stratification, which is not true in a lot of lakes. So this means that you have a higher fraction of lake area in which internal loading is likely to be happening than you would get in other lakes. So there can certainly be differences in different areas of the lake in terms of how reactive those sediments are, but theoretically over much of the lake surface area you can get internal loading. At the same time, Lake Morey is not a very deep lake like something like Willoughby. So that means that when those nutrients are released, they're not diluted over this large volume of water. So they have a relatively large impact on the concentrations of phosphorus in the lake. And at the same time, because you have a relatively thin hypolimnion, so that thin deep water layer, that means oxygen can be depleted relatively quickly, again, promoting internal loading. So what I think is likely happening in Lake Morey over the course of the season is, as Mark and Kelly described, in the beginning of the season, you'll have the onset of thermal stratification, and I represent that here as the difference between the light blue and the dark blue layers. So that'll happen sometime maybe April or May. you'll get these separate water layers develop. As soon as that happens, oxygen starts to deplete. And once oxygen gets low enough, at that point, you'll start to see phosphorus increase. This
Sorry
still
to
is not.
interrupt. You just got a couple minutes left.
Okay, I'm almost done. So you still are not going to see a lot of that phosphorus in the surface water because, as Kelly and Mark said, that's a very stable thermocline during this season. However, once you start to get into fall, into September, even though the thermocline may persist, as you get cool nights, you're going to see convective mixing in the surface water, and you're going to get mixing of some of that phosphorus up into the surface water as the thermocline gets deeper. So over the course of this summer stratified period, you're getting an accumulation of phytoplankton at that deep layer, and that's what you saw in those chlorophyll profiles that you saw earlier. And it's likely that some of those are cyanobacteria. We don't have data on what the community composition of those might be, and it would be good to get some of that. And hopefully maybe this summer we'll get a little of that data. But then once you get mixing, you're also going to mix some of those cyanobacteria up to the surface. So I think that what you would expect to see in a system like Morey was probably more of these late season surface blooms, which is what seems to be occurring. With the few reports of blooms we have recently, they've been August, November, and December, so all relatively late in the year. And this is kind of consistent with the idea that you have these deeper populations developing, which are then mixed up into the surface water later in the year. And this also might suggest that we might not capture all of that with our normal monitoring data, which is generally in the middle of the summer with the lay monitoring program. So this is where reports of cyanobacteria from lay monitors would be particularly useful, I think. So here's a bloom reported last December by Greg Allen. So this was, I think, December 15th. And you can see that photograph of the bloom there. So that's really quite late for a cyanobacteria bloom. So what's going on with cyanobacteria now? We really don't know because we don't have enough data, and we'd like to get that. There has not been consistent cyanobacteria monitoring over time, but we hope to use microscopy samples to better understand the species composition, which might give us a better idea of what might happen if the phosphorus trends continue to increase in the lake. But as a general statement, it's pretty likely, given that there's already blooms reported there, that if P continues to increase, we will probably see more blooms. And I would guess that those are going to continue to be concentrated later in the season. So I'll leave it there.
We have a couple of questions related to this, but I think some of them will be addressed by Danielle's presentation. So why don't we move on to that, and then if not, I'll bring them up at the end.
Thanks, Heather. All right, I will share my screen again.
We see it, Danielle.
All right. Yeah, so I think I might be able to get to some of those salt questions people had. So I'm going to provide you with a summary of the water quality results from the stream monitoring carried out by the Lake Morey Commission, led by Greg Allen, that was supported through the Department of Environmental Conservation. So tributaries are streams that drain into the lake. I think Kelly pointed out a number of these earlier. So these are all the streams that drain into the lake. On the right side of the screen is a map of the Lake Morey watershed and its sub-watersheds, and it's all outlined in black. So the watershed is this area here, and it's the land area that drains to a water body, a lake or a river. In this case, the watershed of Lake Morey is the land area that drains to the lake. And the sub-watershed, like outlined here, is the land area that drains to the tributary, the stream, just before it enters the lake. So rain that falls within these sub-watersheds will either be sucked up by soil or trees, or will end up in the lake, and they can pick up pollutants on their way there. So we want to do everything we can to create environments where It's set up so the pollutants will be kind of sucked up before they get to the lake. So these watersheds include all the land area that drains to Lake Morey, and each of these tributaries is a representative of each subwatershed. Our tributary monitoring started in 2019. Our volunteers have monitored 7 small tributary watersheds on both the east and west sides of the lake. The streams that we've monitored thus far are Glens Falls Brook, Big Brook, Garden Side, Aloha Manor tributary, Bonnie Oaks tributary, and Pine Brook. So these are pretty similar to the sites that Kelly was talking about she was monitoring as well during, I think, either the lake assessment and then also the spring sampling as well. So this takes place through June through September. The goal is to get 10 sampling events with at least 2 high flow events. So high flow event is just basically trying to catch the water as it's rising from a rain event. So that helps us to maybe capture some of the runoff and the impacts from runoff. So as you can see, most of the watershed is forested, and as Kelly pointed out, most of the developed land consisting of roadside and shoreline development, and it's densest along the shoreline. And then we also, I think what's really important to point out is that 2 of the tributary watersheds to the east, both Bonnie Oaks and Aloha Trib, include the Interstate 91. And Pine Brook doesn't. So it makes for a really good comparison case when you look and compare Aloha and Bonnie Oaks to Pine Brook, because they're all kind of on the same side. They're similar types of watersheds, but Pine Brook is the only one without the interstate. So we're heading into a 3rd year of monitoring where we hope to get more flow events to understand those high flow contributions where water's actively running off the land, off of driveways, off of rooftops, off of old forest roads, etc. And the phosphorus concentrations are the highest during flow events in all streams. So where we have captured flow events, we find that that's when we see the highest amounts of phosphorus. So we know, and this is pretty common, so this isn't crazy. We kind of expect this. It really comes down to how high those levels are, especially when you compare a developed area to a non-developed area. What we've also seen, and similar to Kelly's sampling, is that Pine Brook and Bonnie Oaks, followed by the Aloha tributary, have the highest phosphorus concentrations on the average. So that's where we're seeing the most. And I just want to be clear, these levels aren't alarming. They're nothing like some of the agricultural watersheds that we see, like, up in the Missisquoi or anything like that. But they are elevated and they're different compared to what we're seeing across the lake. Chloride and sulfate levels are elevated, and also the manganese, like they talked about earlier, in Bonnie Oaks and Aloha. So I just want to make that point that Aloha and Bonnie Oaks, the 2 watersheds that have the interstate running through them, have the elevated chloride levels. So I saw folks were concerned about the road, the ring road around the lake. And while we can't really monitor what's coming directly from the ring road where it's not, coming into a tributary, what we have seen, at least for all the tributaries that are not connected to the interstate, is that the levels in those tributaries range from about 3 to like 13 milligrams per liter. So it's really low as compared to what we're seeing at Bonnie Oaks and Aloha tributary, which can range from 141 to 200 plus. So that's magnitudes higher. So when it comes to the salt, we think that the interstate is definitely a good source there. And also Big Brook and Glens Falls have the lowest concentrations of all parameters monitored. So that's what we've seen so far. We really hope to do is be able to get some runoff events and see if those levels climb more significantly. So we're doing additional monitoring on Bonnie Oaks to understand the nutrient and chloride sources in that area as well. We've also been working with our geology staff to understand how bedrock geology may be contributing minerals and nutrients, and if it is. And one important note is that tributary monitoring does not allow us to understand the contributions from the developed shoreline area where they don't flow into the streams first. So this whole shoreline here, there's no like tributary that captures it that we can say, this is what it's contributing to the lake. So that's why it's really important where we can reduce runoff along shoreline properties that we do that, because it's really difficult to measure what those contributions are. So that's it for the information for our monitoring from our stream monitoring. And while we continue to monitor water quality to understand trends and potential sources better, we think that the best next step for Lake Morey for the watershed is to embark on a lake watershed action plan. The monitoring helps to inform this plan. And these plans answer questions like, what issues threaten the health of our lake most? And what can we do about them? And I think somebody had a question about that. Where's the phosphorus coming from? What can we do about it? And that's what we hope that the lake watershed plan will help us to answer. So what is a lake watershed action plan? It's an exercise designed to identify, summarize, and communicate the problems, the water quality problems we see, and also the fixes. They identify fixes within the lake watershed that will help to best protect the clean water, the wildlife habitat, and the lake's ecosystem. And it's a community collaborative effort, which is one of the things that's really kind of exciting about these action plans. So our department has funded 5 of these to date in Vermont, and that includes Lake Fairley. So they're just getting theirs underway. And the Lake Champlain Basin program are funding another 4. And we think because what we're seeing with Lake Morey's increasing phosphorus totals, the presence of intermittent bacteria blooms, we think that Lake Morey is a really good candidate to do this work, and it can help to identify where sources are and what can be done to address those sources. To give you a status update on that, our department has issued a request for proposals to hire a service provider that can develop a lake watershed action plan for Lake Morey, and that would start later in 2022. And like I said, this is a collaborative effort. This isn't something that the state kind of does on its own. This is something that the state and partners will work with all the watershed folks. It really is a really good collaborative effort, so you can be involved as much as you want. There is an informational meeting on this subject that's scheduled for March 23rd at 5 p.m. and we can provide more information about that. So to wrap up the presentation, we wanted to close with recommendations from our DEC scientists and our planners based on the information we've collected up to this point. So we recommend let's continue doing the monitoring, and that's the monitoring that the volunteers are doing, and let's support those volunteers in that monitoring the best we can, because that will help us to track changes over time. We recommend community participation in lake watershed action plans, so get the word out and get involved, and then once the plan's been developed, work hard to implement the actions that are identified. And then also just ongoing assessment and implementation of lake-wise at existing shoreland properties. And the thing that's important and really great about the lake-wise program, which if folks want to learn more about that, that's something that we can do, is that we focus on localized issues. So you can do an assessment of your land, and that assessment can tell you the things that you can do to improve it. So it's when they happen, it's identifying sources and solutions. So while our department will continue to support the Lake Morey watershed community through monitoring and lake watershed action planning, we encourage the community to feel empowered to envision what you want your lake watershed to look like in the long term. And remembering those 3 C's, the community, so your community involvement and visioning for the future of Lake Morey, the commitment, and that's something that we've seen with this community as well, is implementing practices that improve water quality and protect habitat, And then continuity, so making sure that you're really building on generational stewardship, educational programs for future generations. And the lake will reflect the efforts that have been put in. And I think then you can really be sure that the community has done their part in protecting water quality in the lake. And so this is the end of the slideshow. I'm going to put the survey in the chat, and then we can open it up for questions, and I'll close it out.
Hey, Danielle, do you want to, I've got I guess 5 or 6 questions that some of them we've answered, some of them maybe we want to return to. Do you want to address those first?
Yeah, that sounds good.
Okay. So the first one was from Laura Tucker asking about, is the road salt from Ring Road or from I-91?
So based on the water quality monitoring, what we're seeing is we're not seeing a lot coming in through the tributaries. We can't say exactly what's coming where Ring Road empties in from the shoreline, so it doesn't go into one of the tributaries, but we're definitely seeing way higher elevated levels in the interstate watersheds. So of course, the town can look at and say, are there areas where we see like more runoff, you know, that we can capture, or where there's erosion happening? Also, if the town salts the roads in the summertime, thinking about, are there different opportunities for that? But I think overall, probably the majority of the salt is coming from the interstate based on the water quality data we have. And I don't know if Kelly or Mark feel differently about that.
No, I would agree with that. Pretty high numbers from the interstate, which I think was, I mean, from the study in the 80s, kind of found similar things, especially with the sulfate as well. So it could have been from some construction of the interstate and road salt.
And we have been in conversations with the Agency of Transportation about their salt plan. So we definitely will continue to have that conversation and see if there's things that we can do to help with that input.
As a follow-up to that, Bruce Duran asked, has Fairleigh done anything to reduce the use of salt?
I don't know, Tad, do you know?
Yes. As we continue to use salt, our road foreman has had a mixture of more sand than salt. So it hasn't been necessarily a reduction, it's just been an adding of more sand. And he has not, I guess you could say he's reduced it, but not significantly. But one would say, if you look at it, it would feel like it was not, because of the mixture of the sand that was there. Hopefully that's helpful.
Thanks, Tad.
I did ask Michael, our road foreman, specifically to that, because I think it was Smith who asked me, not just too long ago. He was very happy to see the reduction. Let's just say Michael has been more, let's put it this way, that you need, in order to get the melting, you have to have vehicles on there as well, believe it or not. So the areas by the fire station and the strip between the interstate and down through Fairview has a little bit more salt because of the highest traction or traffic, if you will. But the ring beyond that does not get as much as those specific areas. Okay.
One other question, Chris Brimmer asked, are there any specific regulatory recommendations that we can implement locally? And then I'll call on Greg.
I don't know if anybody wants to speak to that, but I think that there are the Vermont League of Cities and Towns has stormwater like bylaws, so model bylaws that you could look at. So that's something that could help in terms of, new development and just making sure new development is treating stormwater effectively. I think one of the best things you're going to be able to do is looking at the Lakewise program, and maybe having folks in the community trained on how to do that work, and really have shoreland owners buy into that. And then I think the road erosion, the municipal road general permit also applies to the town, so that should help deal with some of the road erosion issues that come up. And then in terms of private roads, that's another thing that, we don't have any like regulatory hooks for that, but that's something that folks who have private roads is just making sure that they're maintaining those roads. And just identifying when you see that there's a lot of runoff, and determining if there's a solution you can come up with. Because the best thing to do is to work together. As long as you have people who are willing, you're going to get better results, I think. And then there's also the shoreline regulation that the state already has. So I don't know if you want to add anything, Kelly, or.
Yeah, I just would add, I don't know if you have in your zoning bylaws at all, any kind of, 50 or 100-foot buffer requirements on streams in town. But that is something that is under local jurisdiction and would be something to consider. And even though we're not really seeing, with the overall mapping that was done for all the tributaries draining, it was very little impervious surface currently in that narrow buffer area. There is some there, and there is really nothing preventing, future development from building within that very critical area along the streams. So that could be something that you consider, adding to your zoning regs. We do have the Shoreline Protection Act, which protects the 250-foot around the lake shore and has guidance on, what you can and can't do within that area and the permits you need to get if you're going to try to expand a camp or you're going to put a new camp up that would need to meet standards so that would be going in the most lake-friendly manner. But we don't have that for streams, so that's at the town level.
And I do want to call on Greg, but it looks like Oliver has something to contribute to this point.
Yeah, I just wanted to add one point we haven't discussed much today, but I know is an issue down on Morey is, aquatic invasive species, nuisance aquatic vegetation. There is some regulation about this. The state has a aquatic nuisance control statute, but I think more important than the regulatory language is, the preventative measures that Morey is already taking that the state is, continuing to provide some support on around reducing the spread of aquatic invasive species. The clean drain dry effort at the access areas, and really working to keep new invasive species out of Morey and not transport whatever is in Morey already to other water bodies, while simultaneously trying to reduce the spread of milfoil and other plants that are already in there. So that's an important effort that didn't come up too much today, but something that we're continuing in addition to all the water quality monitoring work and planning work in the watershed that we've been talking about today.
Thanks, Oliver. Greg, why don't you go ahead?
Thank you. First, excuse me, Oliver, we almost made it through a whole meeting without talking about milfoil. But I wanted to thank everybody very much. This is just a superb opportunity to talk to everybody and listen to you folks. So thank you very much. And number one, Mark, we did buy that depth meter that you had recommended. So that's ready to roll as soon as we get out on the lake. Thank you very much for your recommendation. And my question is this: listening to you guys talk about the presence of manganese and iron in the hypolimnion, which is anoxic and has always been anoxic to my knowledge, I'm wondering and worried that the work that was done in 1986 with the alum, I thought that formed an alumina phosphate flock that could not be broken. And I'm wondering if the anoxic hypolimnion is having an effect on that material in the bottom of the lake? And could that be a contributor?
So I could speak to that first. I mean, the first thing to know about alum treatments is that they're not forever treatments. And in fact, I think even a goal of lasting about 10 years is pretty optimistic. So it's actually been really impressive in Morey, going on 30 plus years. And so part of that is because, well, for one, I mean, sediment, keep loading into the lake and will kind of cover up the flock layer that was formed from the alum. So you're kind of building, increasing sediment, organic material. And I think over time, just I guess that flock can be weakened. I don't know if others want to speak more to that, but yeah, as far as I know, I mean, it's not gonna last forever. And you're still having inputs from the watershed, and the sediment itself, I think, probably has a lot of organic matter, and the phosphorus can actually sort of travel through the sediment in a way, become bioavailable. So there's a lot of complexities in the sediment itself. You'd have to do some sediment sampling to kind of understand how mechanisms are working, but yeah, the alum treatment is definitely not meant to last forever. But comparing the numbers to what they used to be, it's still a lot lower, so being pretty effective.
Thank you.
A couple other questions in the chat of Arlene asked, do you know if cyanobacteria overwinters on the underside of the ice, or does it go down into the sediments? Is Peter still here?
Yeah, I'm still here. So yeah, generally speaking, they do overwinter in the sediments. And I think it's very likely that that's happening in Morey. They can also bloom under the ice. We don't have any data to know whether that's happening at Morey, but it certainly has been observed in places. But for the most part, yes, they overwinter in the sediments.
I think sometimes too the oscillatoria shows up like almost like blood red underneath the ice sometimes. So if you ever see that, make sure you report.
Yeah, and that Planktothricter oscillatoria is again one of those species that tends to bloom in those deep chlorophyll layers, which we see in Morey. So, I don't know if any of you have seen something like that. I'd be very curious to know if you have.
And Noel had made the note, and I think Danielle had responded to it, that the shoreline regs do not include the other side of the road, so the east side of the lake that's within 250 feet of the lake. I think that's, let's see, the shoreline regs do not include the other side of the road that's within 250 feet of the lake. And I assume, is that because it's a federal highway?
No, that was when the act was being passed and Senator John Rogers was on the committee. He got that kind of loophole in. We were modeling, Vermont was modeling its regs after Maine's because we had evidence that that was working there. They don't have that kind of exemption to my knowledge. But to get the law passed, I think they had to concede that to get Senator Rogers to vote for it. So he's not in the Senate anymore if you want to try to get that loophole closed.
So we're at 5:32 and we said we were going to end around 5:30. Are there any final questions? Danielle had put the link to the survey in the chat. Any final questions? Otherwise I'd go into the chat. Yeah, go ahead.
I don't have a question, but before you go, just on behalf of the town, as the town administrator, this has been extraordinary, and we've had the privilege of having your colleagues from VTrans to other agencies of A&R visit with us. So this opportunity has been stellar, and thank you for putting it together.
Thank you, Ted.
Yeah, thank you all for being here and being a part of it.
Yeah, and keep your eyes out for the Lake Watershed Action Plan, so you can all be involved. Thanks, everybody.
Cheers. See ya.
Have a good night.